brief introduction about you. Okay, so okay, good morning, ladies and gentlemen. So I'm very glad, honored to invite my former PhD student, Dr. Aaron Lin, actually to give invite online talk about this kind of special kind of single cell analysis technology. So that actually Dr. Lin actually got a PhD from Hong Kong UST Bioengineering about almost 9 years ago, and then I think when he first went to like ASM, one of a public company in Hong Kong, work on packaging, and then BioLab, right? BioLab also a very big biotech company, and then work for Illumina. Illumina is another world-leading company making the DNA sequencing machine, and then of course they don't call this kind of the chip about, they call the flow cell. From my point of view, it's some kind of microfluidic device, and then he worked for Illumina for I think almost 4 or 5 years, and then so that eventually several months ago I was informed by him that he joined 10x Genomics. There's another very promising biotech company who went to IPO in New York stock market last year. So that without further ado, let's welcome Dr. Lin to give this online seminar about this interesting new frontier of the research in biotechnology. Okay.
Thank you. Thanks a lot, Dr. Lee. Really appreciate my period within in your group. At the time, I complete my PhD project, and more importantly, I acquired very I think good skills in terms of the microfluidics, the single cell experience, together with some presentation skills, which quite benefit my career in the past 9 years. Really appreciate. Good morning, everyone. Today, I'm very glad to present on behalf of 10x to present our technology, our solution, and discuss with you some very promising applications that we can leverage this microfluidic space single cell solution. I think it's a very flexible discussion. So if you have any question, please don't hesitate to raise your question, and we can discuss anytime. So firstly, on this page, you can see all the offering from 10x. So in the biotechnology industry, we categorize different company as the upstream company, like 10x or Illumina or BioLab. We provide the instrument, reagent, and the solution to our users. And there are also some companies we define them as the midstream. It's like the Novogene, the BGI. They provide the service. They purchase instrument from us and provide the service to the end users. And the downstream is some end users like hospital, academia. People focus on some scientific project, leverage this advanced cutting edge biotechnology platform. So as upstream solution provider, 10x provides the instrument, as you can see on the top left. And we also provide our microfluidics chips. Different chips can help us with different applications, like the single cell, like the gene expression, the protein expression, etc. And on the top right, you will today I also will introduce another portfolio from 10x. It's the spatial transcriptomics solution. Different to single cell, which is a microfluidics based. This spatial, we can say it's array-based technology. And on the bottom left, you will see different color, means different applications, like for the DNA testing, RNA testing, etc. And another toolbox, here we see the software. So as a total solution, yeah.
By the way, can you use some like a pointer function to would be easier for, yeah.
Yes.
Yeah, okay.
Okay, now you can see my laser point, great.
Okay.
Yes. So analysis software, it's based on the Linux system, the command line, to help us process the primary sequencing results into some ready to analyze data. And the last software can help us visualize the single cell data. So now what you can see here is a typical single cell results. It's using the t-SNE or the UMAP technology to map the single cell data onto this two-dimension graph to present. So this is solution from 10x. So we provide the instrument and the reagent in the library preparation step. So here what I show you is a complete workflow of the single cell sequencing. That means at the beginning, we need to prepare the samples. We need to isolate the cells from the biopsy sample or from the patient blood. And we prepare the sample, the single cell suspension solution. And then we will use 10x solution, microfluidic based, to prepare in the sequencing field, we call it a library. The library is ready for the sequencing. So sequencing, we know that Illumina is the sequencer giant. Also, we can support, for example, the MGI, the Oxford Nanopore, and the PacBio, different brands of the sequencer. So this is another topic. Maybe we can cover a little part in the following slides. And lastly is the data analysis. So on this page, what you can see is since the first product from 10x, we, at the very beginning, 10x, the birth of this company was focusing on some sequencing, to address some sequencing issues. We know Illumina is a short length sequencer. 10x, at the very beginning, they launched this solution trying to make Illumina sequence some longer reads. And then in 2016, our founder and the chief scientist, they realized single cell field is very promising. So they enter this single cell field by leveraging the same, the very similar microfluidics based microchips. And then we expand the single cell solution from the RNA testing to the protein testing to the DNA testing and some epigenome study. So now we expand from single cell transcriptomics to the single cell multi-omics. And this year we launched an automatic instrument, which is very big, but it can help the users out of this very laborious pipetting step using some automatic liquid handler to help the biotech or the farmer users to prepare very robust samples. And we also further upgrade our technology to higher sensitivity to help users to detect more single cells or to get higher resolution. And also provide another very exciting product to further detect the multi-ohm information. So as you can see, the trend is from single cell RNA to the single cell multi-ohm, which is a trend of this industry. And on this page, it's a very simple comparison. If we just sequence a sample in the conventional way, that means for example, we take a tissue sample from the patient and we treat this sample and put it into the sequencer. It will generate just one data point. The gene expression is the average from thousands or tens of thousands of cells. So what we can say is just the average. So the problem is we know that the tissue or for example, the tumor sample is with very high heterogeneity because it consists of many different types of cells. For example, the tumor cells, the immune cells, etc. And using the single cell technology, we can expand this one sample into a very comprehensive view. We can tell different type of the cells based on their gene expression marker. And we can further tell the number and the proportion of different cell types. So which give unravel a very comprehensive view for us to learn the heterogeneity. So this is the key value of the single cell technology. So in order to help users do the single cell experiment, we provide an instrument. But this instrument, to be honest, because I'm also with the engineering background, so I can see this instrument is very simple. It's actually very simple. The key function of this instrument is to generate some negative pressure. This negative pressure will drive the formation of the droplet in oil structure. I believe you are very familiar with. So this instrument is simple, but it's very robust. So I think as a commercial product, it's very important. So we sell the instrument to the users. They can trust this instrument because there's no any laser in the instrument or any fluidics structure. So it doesn't require very strict, for example, the alignment, the validation step. So the users can easily take this instrument to any lab to do the experiment. And once they see this instrument fail, we just directly replace the instrument without need any engineer to fix it, because it's very easy. So in this instrument, you can see we have a cartridge. We can put the microfluidics on this holder, and push it back to the instrument, and just simply click start. It will run around 18 minutes of the process to generate the droplet. So within the whole library preparation step, you only need 18 minutes on this instrument. All the other time, you will use the pipetting, the thermocycler, the magnetic plates to complete your step. So each run, we can generate 1 to 8 samples to generate, because we have 8 channels, as you can see here. From each channel, you can harvest from 500 to 10,000 cells or cell nucleus from each channel. So this is the throughput of this instrument. As I just mentioned, we also launched an automatic instrument, which is much bigger. So we integrate the previous smaller one here together with some pipetting handler, the computer for these walk-away steps. So only needs 1 hour hands-on time, and all the other time the scientist or the operator, he or she can just walk away. Let the computer control the instrument, complete the step by itself. So yeah, different components. So I won't go into detail. And now this slide is very important, but as a microfluidics researcher, you will find this structure is really simple. It's really simple. But I think a simple way, it also has its advantage because it's simple, but it's very stable. This is very important as a commercial product. So the step comes from we prepare the gel beads. This gel beads, in my opinion, is the most important value or the important pattern of 10X. Here I use different color to label each individual gel bead. And each gel bead is different because on the surface of the gel bead, it has some oligo. The oligo has different ATCG sequence. So from the other inlet, we can input some cells or the cell nucleus. And in this microfluidic channel, they will meet with each other. And in the interface between the oil and the liquid, you will generate a bubble or a bubble in the oil. So the inner part is the water, outer part is the oil. So it's not bubble, it's a droplet to be more accurate. Within each droplet, you will see most of the droplets, it will contain 1 gel bead. And for small part of the droplets, it will also contain 1 single cell. So here we can see 1 single cell exist with one gel beads within a droplet, and then we lysis this single cell and lysis these gel beads. So all the mRNA from one single cell will be labeled with a special barcode, which was exist on the surface of the gel beads. So in this way, we can label each single cell and label every mRNA fragment from each single cell. So here we use different color to label. After that, we break all the droplets and mix all the fragments together and then we send it to library to complete the library preparation and send for Illumina to for the sequencing. And after sequence, because each fragment with a barcode, and we can categorize different fragment into each single cell. So in this way, we can study the gene expression of each single cell. So this is the key idea of this single cell technology.
Sorry, Dr. I want to know that, so in your company product, do you have your own DNA sequencing machine?
We don't have. We don't have our sequencer, yes. So basically we are the library preparation company. We prepare the library and the user still need to rely on Illumina, MGI for the sequencing. Yeah.
I see. Okay.
And here I show you one picture of the microfluidics. So in the middle row, we input the samples, and this row 2 we can input the gel beads, and in the front row, the first row, number 3, we can harvest the formed droplets from this row. And another very important feature of our technology is our cell capture efficiency is up to 65%. That means if you input 17,000 cells per well, you can harvest 10,000 cells from this well. So this is very important because we know that single cell is not unique to 10x, because many other providers also have single cell solution, like the Bioret, like a Fluidigm, like some smaller company in a startup company in US. But 10x did quite well in terms of this capture efficiency. For the other vendors, they only have maybe 10% and maximum 20%. But that is a problem, because if you like to study some very sparse, very rare cells, for example, the fraction is just 1% or 0.5% in the total sample, this 20% capture efficiency, you will lose your target. But this 55% is quite enough. So this is one key advantage of 10x platform. So here, another thing is doublet rate. Here, I will talk about this problem. This issue is not a problem. It's an issue existing in many single cell platform, but I will talk this a bit later. So here is the feature of our microfluidics system. We have a different letter here to label on different microfluidics chips for different use. For example, the gene expression, for example, the protein expression, something like that. Okay, now what you can see is a photo under the microscope. We can see this is a droplet. In the middle is a large gel beads. It's a gel beads. Gel beads is larger than the cell. This is a single cell. And then the other part is the water reagent, and the outside is the oil. We have different chips, as I said, because this instrument is very robust. So you can use the test chip just to measure the pressure, to control the pressure of the instrument is normal. When we check the pressure is normal, we can have the confidence to run the real sample in the instrument. So it's very robust. And bioanalysis, bioinformatics analysis is, to be honest, the top challenge so far in this single cell study. because compared to this bulk RNA-seq, for each sample, we can just, we only get an average set of the data. The data is very small, the data set. But for the single cell study, for each cell, we will generate a lot of data. So as a calculation, to study single cell per sample, we will generate around 100 gigabase data per sample, consists from maybe 10,000 cells. So you can calculate how massive is the data set. So we provide the Cell Ranger and the new browser software, both of which are free to download, came from our website for the analysis. And because single cell community growing very fast, so we have some global wide have some users, some single cell scientists, for example, the scientists in New York Genome Center, I think in UCSC, they are very advanced, very experienced bioinformatician. So they develop some third-party tools like the Monaco, the Sturet. These two are very famous in the single cell research community. They can help users to complete some very advanced analysis. For example, to remove the variation between different batch, or they can help aggregate different samples together for the meta analysis, something like that. Very fancy. And in terms in the data analysis, here I would like to show you how we usually analyze the single cell data. So from the experiment, we get some raw data, as you can see from the figure A. So each row is a gene. Each row is a gene, and each column is a single cell. So you will see this matrix will be very, very long and very, very wide. It's a very large matrix. And then we convert the matrix into some dot plot. And here we can do some QC. We can remove some empty droplets, or we can remove some droplet within more than 2 cells. This is something we need to remove in the analysis. And then we need to separate the cells, right? As we can see here, we need to use a single cell to tell each single cell belong to which type. So we need to find some genes. These genes, they are expressed differently in different cell type. So we said they are the marker gene. So we need to find some good marker gene to differentiate these cell types. And then we will use firstly PCA, and then the t-SNE or the UMAP. It's a I think the dimensional method. I don't know how to speak accurately. It's a very component. It's a principal component analysis, and some very classical mathematician to help us downgrade this very highly dimensional data into this 2-dimension presentation. And then we can can separate cells according to their differentially expressed genes as the marker. And for each gene, we can use the heat map to label the expression level of each single gene. So this is a typical presentation of the data, of the single cell data. As I said, it's very important to tell which droplet is empty, right? As I show you, most of the droplet, they only contain 1 gel bit without any single cell. So we can use some statistical way to remove those background droplets. Or we can use some method to remove this kind of the doublet droplet. So because all the cells, when they be partitioned into each single droplet, they will follow the Poisson distribution. So majority of the cells, they are of the droplets, they contain either no or just 1 cell, but there's a very small fraction of the droplet, they can contain 2 cells. So we can use some bioinformatics way to tell these 2 cells droplets and the discard length in our final analysis, because it's not single cell, right? It's 2 cells. Yeah. So this is another topic. If you are interested, we can talk later. So now I will go to the application part. I think you are very interested in how we can leverage the single cell technology for some cancer study. So I pick up some cancer-related publication and products to introduce here. So the first one I would like to introduce is the gene expression. Gene expression is to study the mRNA, for this assay, I already introduced the throughput, the capture efficiency, and the throughput. So I will skip this page. I will show you how we can capture the mRNA from each single cell. This is very important. So as I just introduced, I think that the gel beads is the top secret of all the secret weapon of the solution, because we provide different kinds of the gel beads for different use. For example, for this very classical one, the gene expression gel beads, on the surface of the gel beads, there are millions of the oligos. So if we enlarge this part, we can see each oligo, it has 4 different fragments. So this one is for Illumina sequencing use. And this one is the identity, is the ID of these gel beads. So all the oligos on the same gel beads, they share the same barcode. They share the same barcode. And once a cell encaptured together with one gel beads, so all the mRNA from this single cell will be labeled with this part. So this is how we label each single cell. And UMI is a little special. If you have a molecular background, you will know that UMI is unique. It's a random sequence. So it will help us to capture each unique mRNA molecule. And lastly is the poly-DT. We know that for the eukaryotic species, for example, the human, mouse, plants, or even the fungus, the mRNA of these species with a poly-A, the polyadenylated tail. So all the mRNA molecules with a poly-A tail. And the poly-A tail is complementary to this poly-T. So we can use this poly-T sequence to capture all the fragments with a poly-A tail. So in this way, we can capture all the mRNA molecule from each single cell, use this fragment. So this paper, maybe I will skip on the background of this paper, because if I talk about this disease, it will be very long. So simply speaking, this paper is to study the breast cancer. It's an immuno-oncology paper. So in the breast cancer, after the treatment to the patient, we need to know what's the effect of this immunotherapy. One marker is if we can see more immune cells enter this cancer area, the more the better. That means the immunotherapy can activate those immune cells to enter this breast cancer tissue to kill those cancer cells. This is the basic purpose. So here we have, I think, several patient samples. They with the triple negative breast cancer. After the treatment, we take the tissue samples, prepare the single cell suspension, and run the simple cell assay. So now we can see here is a very classical single cell t-SNE map. Here we identify all the immune cells using the immune cells marker like a CD8, CD4, and a CD3. I think here they are all the T cells, and then we can separate all the single cells based on their gene expression. So we can categorize all the T cells into different categories, because CD4 T cells and CD8 T cells, they have a totally different function in this immuno response. So in this experiment, they demonstrate to us that a single cell method has a very high resolution. We can tell different types very clearly. And for each type, we can see the expression of specific gene. For example, here for this CD8 positive TRM cell, we can see on the right-hand side, we have 8 names of the genes. Those 8 genes, they are highly expressed in this type of the T cell. In the heat map, we can see the bright color. And for example, for this CD3 negative monocyte cells, we can see, okay, these genes express highly in this cell type. So this is a basic results of the single cell. We categorize cells according to their differentially expressed genes. And for each cell, we can further quantify the expression level of each gene. So this is a typical gene expression level. And now let's go deeper. We expand from the gene expression to other dimension. This is what we call the multi-omics study. Here we have 3 examples. Let's firstly focusing on this CRISPR. So CRISPR, as you know, it's a very popular gene editing method since I think also 2013. And nowadays, many companies, they can make, for example, the Merck Millipore, they can provide a very long portfolio of the guide RNA with some specific designed sequence for the researchers or the clinicians to targetedly cut off some genes to inhibit expression of some genes. This is the major application of the CRISPR. We introduce some guide RNA to cut some fragments of the DNA, and in this way, we can either inhibit the expression of some genes or activate the expression of some genes. In this way, we functionally modified a single cell. So how we can help in this CRISPR screening? This is another very smart design of 10X. Here, as I just showed you, we have these gel beads. The first fragment I already introduced to you because we use the function of this oligo is use the poly-DT to capture all the mRNA of each single cell. And then we also have another 2 oligos. They have some very specially designed capture sequence. It's a special sequence like here. And on the other hand, we can use, our users can order this guide RNA from Merck or from other vendors. This special guide RNA, they has one additional sequence here, which is complementary to our capture sequence. That means this guide RNA, as long as it exists within this single cell, it will be captured by our gel beads as well. And on the other end is the special sequence of this guide RNA. This sequence is called the protospacer. So it will determine in which location this guide RNA will enter, will cut. So we can see this part is the identity of this guide RNA. So within one single cell, let's imagine, firstly, all the mRNA will be captured by this sequence. And secondly, as long as some guide RNA within these cells, it can be captured by our another 2 sequence. So in this way, we can build one association, which guide RNA in this single cell, how it change the expression of the mRNA. So this is a smarter design. So as we can see here, we can leverage like Merck to manufacture this guide RNA encapsulated by the lentivirus and transduced or electroporated the cells. And once the guide RNA within the cells, we can culture the cells for 2 or 3 days, and we can categorize those cells according to the guide RNA within it, because each guide RNA has a specific identity. And then in this way, we can further dig in the expression of each cell. So as another example here, it's also a CAR T. I think it's it's a CRISPR gene edited T cell. It's a immunotherapy, innovative immunotherapy study. So this study from UCSF. Firstly, the authors used I think that the purpose of this paper is they would like to try several unknown guide RNA. Why I say unknown is they find this guide RNA may be a potential to kill the cancers, but they still not very sure about which genes will be regulated by each individual guide RNA. So they use our technology for this screening. In this screening project, they transduced totally 48 kind of the guide RNA into the T cells. And they would like, for example, inhibit the expression of the PD-1. Well, maybe very familiar, the PD-1 immunocheckpoint. We need to inhibit the PD-1. It's a classical immunotherapy way. And then they would like to try the other guide RNA. For example, those guide RNA may help the proliferation of the T cell, or maybe they can enhance the effector of each T cell. So maybe we're bringing many different kinds of positive effects to the T cell. So with this hope, they tried 48 guide RNA, and they identified totally 7 guide RNA as shown in each column. And each row stands for each genes. So these genes, they are very important in the function or in the life cycle of this T cell. So we don't need to dig further or which color means. Simply speaking, these 7 guide RNA, they can make some positive effect on these genes and to enhance the function of the T cell. So in this very high throughput way, they quickly identify these 7 guide RNA. But if they using a conventional way to do this CRISPR sequencing, they need a double time, and then they need, I think, because it's just one assay, we just harvest one batch of the cell and do analysis. But in a conventional way, if they would like to try 48 guide RNA, they need to prepare the you are familiar, the 96 well multiplate. They need to at least half of the plate to separate the 48 guide RNA to culture 48 sets of the cells individually, test individually. So it will take a very, very long time. By using single cell way, they can test 48 guide RNA within one batch of the cell quickly and very efficiently. So this is another very important application. And then I will go to the immune cells. So the immune cells, I think especially this year is a very hot topic because we know the COVID-19 pandemic situation. In the vaccine research and in the research on identifying some new neutralizing antibody, like a Professor Sunny Xie, who was working on this project in Peking University, and recently I heard that they already opened a company to develop this COVID-19 neutralizing antibody, and their project are based on 10x. Their project. So this is how they did in this way. So a very simple background is we know that the T cell and the B cell in the human body is very with the massive diversity. We can say you know that in the mother nature we know that maybe 10 billions of different pathogens, like the virus, different bacteria, and many other pathogens. But what a human can survive, because he or she in their body they have equally the same level of the type, the B cell or T cell type. Because maybe, for example, as take me for example, maybe in my body there are also 10 billions types of the T cell or B cell. It's very largely diverse. So why? Because each B cell or T cell on the surface, on the membrane surface, they will have a B cell or T cell receptor. But if we take two individual T cell or B cell, their receptor is different. That's why they can target to different pathogens, different virus. So what makes this TCR, BCR so diverse? Because the genes, they can have a randomly recombination and some super somatic mutation frequency. So it's the genes that make this receptor changes or mutates very frequently. So it brings many diversity. So our technology is, after we get all the mRNA, we can design some primer to amplify those VDJ fragments, which will encode this receptor. We can further sequence this VDJ. In this way, for example, in the human body, we take the blood and we isolate those here, the B cell, the P cell, which secrete the immunoglobulin. We can tell for each individual B cell, we can sequence its a genotype, its amino type, and we can quantify how many cells with this kind of genotype. For the T cell, we work in the same way. So what Professor Sunny Xie did is they identify hundreds of the cured patient or the cured patient. They infected by the COVID-19, but they get healthy by themselves because their self-immune system did work. So Professor Xie would like to know which kind of B cell in the patient's body help the cured from the COVID-19. So the principle is they studied all the B cell from this patient, and they identify some of the clone type with a very high frequency. So they can judge, okay, this kind of the genotype, this kind of B cell is our candidate. So then they did further validation to quantify the affinity between the antibody and the antigen. Finally, they narrowed down, and finally identify maybe 10 or 15 types of the B cell. Then maybe they can serve as the medicine once they inject this kind of antibody or this kind of antibody into the body of the people. They can help the people to neutralize the COVID-19 virus, at least within several months. So this is what they call the neutralizing antibody. So they discover using the 10X platform. And then we even further expand the study of the immune cells. So here is the example. So as I said at the very beginning, we study the whole transcriptome, right? We study all the mRNA within a cells, and then here, as I said, we can study the VDJ sequence. We can know the genotype of the receptor, the BCR or TCR on the surface of a B cell or T cell. And then we collaborate with the other two companies. We can further study, one thing is the surface protein of each B cell or the T cell. And then we can further study the antigen specificity. So for example, we can add some human-made antigen peptide, and we can use this like a bait. We can like when we're fishing, we use it like a bait. we can get some T cell that is specific to this antigen, and then we can further dig the sequence of this TCR, so we can link these two dimensions together. So in order to implement this kind of application, we collaborate with two companies. They designed these two products with us. The first one is the antibody. In conventional way, when people use some flow cytometry to study the cell membrane surface, they will use some antibody labeled with the fluorophore, like the FITC, like the CY3, something like that. So from the flow cytometry or from our microscope, we can tell the fluorescence color, right? Then we can know which kind of antibody exists on the surface of the cell. So the pairing protein of the cell does exist, and we can even tell we can even quantify the expression of the surface protein using the fluorescence signal. So this is a conventional way, but now we can replace the fluorescence fluoro using this barcode, and this barcode consists one feature barcode is a special designed sequence, ATCG. And once in the sequencing results, we can tell the sequence, we will identify which kind of antibody exists on the surface of each individual single cell. That means these individual single cell, they express a protein that is pairing to this antibody. So this is how we quantify the cell membrane protein. And another way is we collaborate with another company, design a very similar structure, but this structure, they are unique to the T cell. They can only bind the surface, the TCR of the T cell. So in this way, we can use this structure to capture all the T cell that is specific to this kind of pathogen. For example, we can design COVID-19 because we know that the SARS-CoV-2 virus sequence. We can design this peptide as a mimic of the COVID-19 virus. And we can capture all the T cell that is specific to COVID-19 and further study these T cells. So this is the expansion, as I can see. The first technology we collaborate with BioLegend. It's a very famous antibody company. The second structure we collaborate with Immudex. This company is smaller. It's a Denmark company, but their product is very, very interesting, very promising. So from each single cell, now we can get 4 libraries, the cell membrane antigen, the TCR, and the gene expression. So 4-dimensional. So we can study each immune cells from multiple dimension to get a very comprehensive landscape. Like here, it's an example. So this is a PBMC, the blood sample from a patient. This patient, as we know, it was infected by the CMV virus. Because using this technology, we find from his blood that there are a smaller cluster of the T cell. They are specific to CMV virus. So that means, okay, this person was infected. And another interesting thing is we didn't understand, we didn't know that he ever got infection by EBV and got infection by flu, a kind of flu virus. But from the results, we designed some specific antigen. We also, like a bait, we find some small cluster of the T cell. That means, okay, now we understand this person was infected by this kind, another 2 types of the virus. So this is very, I think, very sensitive assay. And then in order to better serve our, we call it translational researcher. In this industry, we said, like Hong Kong UST, we are a basic researcher. We study some basic mechanism, and our purpose is to publish paper. But for some translational researcher, maybe they often work in the medical school, or they work in the pharmacy company as the scientist. They would like to identify something new and firstly publish paper and then trying to commercialize this kind of identification as a way of a new therapy method or in a way of the new medicine. So for those translational researchers, their target is very focused. If I just would like to study the cancer, so I only interested in those cancer-related genes. I don't need to study maybe 20 to 30,000 of genes. I only need to study just 1,000. So to better serve this part of the customer, we have the target RNA testing. So it's very simple. We prepare the whole transcriptome of the cells, and then we use some baits or we use some probes to capture these 1,000 genes. Only these 1,000 genes. The benefit is in the following string, because they need to use the Illumina sequencing to study these 1,000 genes. Compared to these 20,000 genes, now they only need to study 1,000 genes. So the sequencing cost will be much lower, just 20, maybe 10% or even 5%. So we can help our users to save the cost. So this is, I think, the key benefits of this target panel. So the target panel, we provide some designed fixed content panel. All the genes was suggested by the top researchers in the cancer field. All the users can use our online tool to customize their own panel. Just input the name of the genes, or the ID of the genes. We can help them design this panel and to lead the users to ITT. ITT is another company whose expertise is to manufacture those oligos. So to make your own panel. Yeah, I think I also did this work in UST. So made this panel and build your own study. Also here we can see, compared to the whole transcriptome, if we use the targets way, with much lower sequencing depths, only 1/35, just maybe 3 to 4% of the cost, we can still categorize the cells into different cell type. And lastly, of the single cell part, here we know, maybe you heard about this epigenome, because we know how all the cells, right? All the cells, they have the same genome. For example, all the cells in my body, for the skin cells, my heart cells, they all share the same genome. But why they are quite different? Because the gene expression and the protein expression is different. So that's why make some cells as the skin cell, some cell as the heart cell. So how to regulate which genes to be expressed in each single cell? This is realized in the epigenome. So in the human body, our DNA will be very diverse, actually. Some of the DNA part, they will be more open. Some of them will be more closed. For this very open region, there will be some transcription factors bind to this open region, and they will send the DNA a signal to drive the expression of the genes. So in order to better understand the mechanism of each single cell, we firstly need to quantify the gene expression level, and secondly, we hope to study this open region. So 10x, we just launched a multimode method. We can, from each single cell, study both this open region and the gene expression. So the idea is also very strict. We can use two kinds of oligos, right? The first one, you are familiar, it's to capture the gene expression. And the second one will help us to capture like this open region of the DNA. So within each single cell, we can make two libraries. One is open chromatin, one is single cell, and we sequence them separately. And in the software, we can link the results together. So here we generate two-dimensional methods. We can, using the results to interpret each other. And finally, on this page is a cancer study. It's trying to study the B-cell lymphoma, lymphoma cancer. And they would like to know why what's the difference between the tumor B cell and the normal B cell. So from the gene expression, they see the tumor B cell, the orange color. The gene expression of IL4R is very high. Yeah, it's the marker of the tumor. But what makes this expression, right? Because they are the same. They was the same cells, but why some of the cells, their gene expression expressed very high. So they identify that, there are a peak here, a very open region here, which was 24 kb upstream of this gene. Because there is very open, so they improve the expression of this gene, which finally leads to this tumor. So from this epigenome view, they can identify this peak. So at a very big, just based on the gene expression, we know the gene is highly expressed, but we don't know why. So that's why we integrate the epigenome view to interpret, to give us a full story of the gene expression regulation. And lastly, I would like to show you another very exciting technology. I won't take a very long time, just several slides. We call it spatial transcriptomics. So this thing is very interesting. Like in Hong Kong, if we would like to invest the real estate, or the Li Jiacheng, he always said that location is the most important if you would like to buy an apartment. So I think in this study, location is also very important. As I said, if we would like to study a tissue, right, a breast cancer tissue, in a single cell way, we just take the sample, we dissociate all the single cells in the suspension to study. We can identify each single cell, but we don't know which location does this single cell comes from, right? We don't have the spatial information, but in this study, in this way, we can identify. Here is a very quick example. So this is two patients. They got the breast cancer and they got the immunotherapy. After immunotherapy, they would like to observe what's the effect of this therapy. So for the patient one, we find all the blue color, they are the they are the cancer tissue, and the red one, they are the immune cells. So for patient one, we see, okay, the immune cells, they finally enter this cancer tissue. I think these immune cells will help cure the cancer. But for the second one, still the immune cells, they are still all located out of this tissue sample. So the prognosis or the therapy effect of patient two is much worse than the patient one. So this is the spatial information can provide to us. So for the spatial thing, it's also very smart. We use the glass slides. The glass slides is commonly used in the pathologist lab. And on the slides, it's very specially designed with some mini dots. Each dot is only 55 micron. The dots, I think the function of each dot is very similar to the gel beads in our single cell solution. So each dot, they have some spatial, some unique barcode. So finally, we get the mRNA from each location, and we mix all the mRNA for sequencing. And finally, we can tell where was this mRNA coming from based on this spatial barcode. So we can allocate all the mRNA molecule into the spatial location. So we can generate these kinds of picture. So let's quickly move to this page. So on the left-hand side, it's a classical tissue sectioning. Right? They use the HNE staining. It's named hemotoxylin and erosin. It's a commonly used stain way by the pathologist. So occasionally, if the pathologist in our, for example, Prince Wales Hospital, they just observe this purple color picture. By telling the morphology, they will give a judgment. Okay, this part is a tumor part, and this blue part should be the immune part. they just make decision by their experience, by studying this picture. So it's not a very accurate way. So using this sectioning slides, as I said, we can identify each spot by telling their gene expression. We can give a judgment. These dots, they are the cancer dots because they're with the cancer marker. And these dots, the deep blue color, these dots, they are the immune tissue because they with the immune cell marker. So our results has a good concordance with this pathologist judgment. So, but this is a good way. But in more cases, we can see our method is more accurate. We can tell some sub area with more solid proof than the judgment from the pathologist. So this is, I believe it's very promising methods. In the future, we can quickly enter the clinical field. And besides this H&E staining, we also support this immunofluorescence staining. It's like using the microfluidics, we use some fluorescent antibody, as I just introduced, to study some protein expression. And now we can integrate this protein study together with our transcriptome study to merge two dimensions together. So on this picture, we can, for one thing, we can identify which kind of protein expressed in this tissue. This is, I think, the brain tissue of a mouse. And on the other hand, we can know all the gene expression of each dot. So yeah, now I think it's the end of my presentation. Thanks for your patience. And now we can have some free discussion. Thanks for your time.
Okay, thank Dr. Lin's presentation. So that I don't know if do you any of you have some question for Dr. Lin? Well, I have some quick question for you. So yeah. That according to what you present your presentation in the introduction slide, you say your cell capture efficiency of your product is about more than 65%. So my question is, how about for those kind of it is good enough, and I don't know if this kind of efficiency has impact on performance of the result.
Oh yes, I think
if there's some way to increase to even higher, 80% and 90%, what happened to the result of this this kind of single cell analysis result?
Yeah, it's a good question. So firstly, we can say this number is so far is good enough, but it's always there's always space if we can further increase this capture efficiency. Suppose, for example, as I just introduced, if we would like to study the B cell or the T cell, so every single B or T cell, we can say they are unique. They have different TCR or BCR. So if our capture efficiency is still 65%, we will still miss the 35% of the loading cells. Maybe there are some unique TCR or BCR in that part, but we missed. So yeah, so it's always good to further increase this capture efficiency. But now let's in terms of technology, it's kind of a dilemma, right? Because in single cell study, for one thing, we are afraid that we lose too many samples. In another way, we are more worried about this duplicate problem. Because if we capture efficiency is very high, but on the other way, there are more fraction of the droplet. They may contain 2 or more cells.
Oh, I see.
Yes,
I see. So that if you increase the efficiency too much, and then sometime okay, I understand. So that means so each droplet has has a problem. I understand. Oh,
yes, the multiplate rate. So here, our suggested range is every well you can load 800 cells to 16,000 cells, but you can tell the multiplate rate also increase with the loading. So that's why we don't suggest our users to load more than 16,000. In that way, the multi-rate, once it above 10%, it will bring in the challenge to the results.
Then my question for your company product is this one require a lot more detailed engineering of microfluidic. This is now design. So that you trade-off between capital efficiency and you can see this kind of is expecting multiple rate. So that I think it's possible to improve, but that requires a lot more detailed engineering about this product.
Yes. Yeah, exactly.
I see.
I think also as a commercial company, I think our R&D team, they also care about the cost to make these microfluidic chips. So I think they got a balance between the performance and the cost. So finally, they reached to this 65%.
Because I do, although I didn't work on drop-based microfluidic, but I do monitoring this particular field for many years. I do understand the basic principle and also the critical design parameter for this kind of drop-based microfluidic, so that require additional, for example, additional engineering the surface interfacial tension.
Yes.
Droplet and two droplet everything, and also surface hydrophobicity. Now you need additional surface modification about your microfluidic device. So there's a lot more detail to be engineered so that you can really improve this kind of basically in real final product. You need to engineer multiple objective of your performance, so that that require additional. As I understand.
Yes, and I think one top secret, we as the commercial team, we cannot never ask for is what's the formula of this oil. I
understand. So that's relate to some of your proprietary kind of technology. I understand. Another question I asked is about at this moment, I know that your company is really only make a microfluidic device, and then additional key technology was like gel bit, this kind of formulation, and then so that so at this moment, your company only work with Illumina sequencing like a machine, and also the BGI's the instance the machine. Also, what's the current your product portfolio?
Oh yes.
So how many DNA sequencing machine your product can be together?
Yes. So theoretically, our solution can be compatible with all the sequencer, all the major sequencer provider in the market. But Illumina is the most user friendly because we already integrate Illumina sequence on our gel beads. So this is Illumina sequence. So it's seamless integrate with Illumina workflow. But we also have several users. They use MGI. For example, the Professor Zhang Zheng from Shenzhen Third Hospital, he just published COVID-19 papers with very high impact several months ago. And in their paper, they because MGI, they provide another kit. Using that kit, they can use PCR to replace Illumina sequence into the MGI friendly sequence. So with an additional step, change this sequence to MGI sequence, they can use our library to sequence on the Huada or MGI sequencer.
Okay, I see.
Yes. And also for this long read sequencer like Oxford Nanopore and the PacBio, we can also support. But we not officially support, but our users can always change the step to use on that instrument.
I see. Of course, your common product is primarily try to be compatible with this kind of mainstream DNA sequencing companies' product like Illumina. So that these company or Huada or BGI usually they use optical, right? Optically do this kind of sequencing. Then my question is, my team, we do in my own lab, we are actually working on this kind of a semiconductor. This kind of sequencing. Electrically, we try to see can we use TSMC, CMOS foundry, or CMOS or CMOS main foundry to look at electrically DNA sequences. So that I just wonder, is possible we may try to learn from your company products, think how to be compatible with the company product. What do you think?
Actually, yeah, I can share with you some information from Illumina, because I just left Illumina. It's not a very top secret, actually. Illumina,
I know that Illumina also working on this semiconductor.
Yes, Taiwan,
yeah.
IC model. But for our instrument, we don't have any optical. We don't have any optical structure used. Why? Because Illumina sequencer, they will, I think, let's using this for example. For example, if in Illumina sequencing, every time they add one base, they're using an optical way to detect. But in our workflow, we didn't need to use any optical step, because all the sequence, they are already confirmed. We don't need to know the unknown, because that part we will rely on Illumina sequencer. We just prepare this mRNA of each single cell, but what's the sequence of the mRNA? We don't need to care, because Illumina sequencer will give you the answer. They will leverage the optical way.
I see. So that means I can see this, your GeoBeat is really one of the most critical like invention, enable a lot this kind of application, right? From my point of view, the more things. First is your new idea of GeoBeat, how you have this kind of barcoding and now integrate this kind of your microfluidics, so that you can really do this kind of new type of single cell like analysis. I see.
Yeah. Do you mind if I open another?
Oh, no problem. No problem. Yeah.
Let me open another slide. That there is a slide very clearly shows with. Let me see. Okay, here. Yeah. Let me share the other one. Yeah. So once our technology, we are the library prep. So we complete this preparation of this, for example, these fragments, and all the color labeled part already with the known sequence. So because we designed, so we know all the sequence here. Illumina sequencer will give us the sequence of this part. This is something unique, something they need to what we need to do sequencing, because we need to know this part.
I see.
Yeah. So when in the Illumina instrument, they will add sequence the ATCG base one by one. Once they add one base, they will use optical or semiconductor way to detect which type of base, A or T or C or G. So they will know the sequence of this part.
I see. Yeah, I think this is a very, it's really a new degree of freedom to work on this kind of sequencing or single cell analysis from different, very different angle. I see. Invention. Yeah.
And I think Oxford Nanopore, they use quite a different way for the sequencing. They detect the electrical signal.
Yeah, using the nanopore, so that without any labeling, then they can actually they have spent more than 10 years to overcome this kind of noise issue. Yeah. So that finally they can really make work. I see. So that your company have some way to make some additional new product to be compatible with Oxford Nanopore.
Actually, it's compatible. Not a commercial. I think we won't launch a commercially available kit. But when the users would like to use the Oxford Nanopore, I think because all Oxford Nanopore, they are a long length sequencer. So they using Oxford Nanopore, they don't need to do the fragment here, because Illumina is a short reads. We have to cut very long fragment. But if the user using Oxford Nanopore, they won't do the fragment, but they will use other way to add this adapter on the both sides. So they will generate a much longer fragment in their sequencer.
I see. Very interesting. Okay, I don't know if we have some question from the other student or other people. They have any question? So of course, one more question I also ask.
Can I ask a question?
Okay. Yeah.
Oh, sure.
Yeah.
Just one question. So, in your opinion, what's the advantage of this droplet-based microfluidic technique over the microwell-based microfluidic technology? Because I remember you mentioned that droplet-based can achieve a 65% single-cell accuracy. Mm-hmm. And you also mentioned that, for example, the BD, they use microwell-based. You mentioned that they can achieve probably 20% or something. Is that right? Mm-hmm. But actually, there are some other improvement. Other teams, they can achieve more than 90% or even 95% accuracy. If they can achieve such higher single-cell droplet, then I can't see any advantage of droplet-based compared with microwell-based. So what's your opinion?
Yes, so to be honest, suppose we have two perfect products. One is droplet-based, one is microwell-based. We cannot tell which one is better. But we can only say at this moment, two companies, our existing products, we have two different ways. So far, we have some advantages in some key performance. The first one is their microwell, they also face this dilemma because the size of each well, they need to make as one size. Now, so now their well can only be compatible with the cells smaller than 20 micron. 20 micron because the limitation of their well size. So if a cell is too large, their microwell-based capture efficiency is much lower. So our platform, we are compatible with 50 micrometer. So this is the difference of the instrument design. And secondly, we have some better, I think, competitively advantage in terms of the assay. Now we provide not just the gene expression, but also, as I introduced, the VDJ, the ATAC, or different assays. So for all the assays, we use our droplet, use our gel beads. But so far, BD can only support this gene expression. So there are, I think, for the market share, BD is smaller, but we also see some customers, they like BD quite much because they said 220 micrometer is quite enough for their use. And they only need to study the gene expression, so they buy BD. But once they would like to study the other assay, so they say, no, we cannot do on the BD platform, we need to use another one. So yeah, this is my answer, but we cannot say which method is better. They are both very good methods, to be honest. Yeah.
Okay, I have one more question for you. It's about the last part. You demonstrate it's kind of spatial way to look at, for example, tumor, right? You look at single cell, and then how, I don't understand about how you can map this kind of, okay, now maybe make a doctor cut one part of this tissue, right, from the tumor, and then still I still don't know. So spatially you label each cell, and then how you, of course, once you get the tumor, and then you have to break the tumor into the cell suspension. So still that part still cannot catch the way you can really have interesting way to map back to the spatial resolution about this kind of thing.
Yeah.
That part is quite amazing to see the way you present it in that slide.
Yeah. Do you mind? Let me open.
Because I believe this is will be quite important, useful for us to understand more on this kind of how the tumor, right, primary tumor can be become what metastasis, right? You mean once you really can check those single cell of one tumor, and then if you're able to do the labeling about every cell, of a tumor, and then also link, or maybe some will leak out through the CTC or cancer stem cell. Then this kind of your company product has provide a very good this kind of technology to understand this kind of cancer in much deeper level.
Yeah, yes.
Yeah.
Let me spatial. Let me show
That's
another.
really amazing from my point of view. Yeah.
Let me quickly share another slides. I think it's a training slides. We will give a comprehensive training for the use. Let me see if I have another slides. At the beginning, I think first of all, we cannot use spatial for the CTC because CTC is they exist in the blood. So
I'm interested to understand about this kind of mechanism, right?
Yes.
Once your primary tumor, so that how use your company product.
Yes.
To really do the checking.
Oh yes.
All this kind of cell in the tumor, right? And then to see maybe some of those cells in the tumor will turn into the CTC.
Yes.
That means I believe your company product has this ability to the tracking.
So, for the, I think for the tissue sectioning, we will cut the tissue sectioning as I think the thickness is 7 to 10 micron.
Okay.
So then we will firstly fix the tissue. So we will fix all the cells, and then we will permeabilize the cells. That means we will jig some pores on the membrane of the cells. All the mRNA will fall down. By the gravity fall down on the bottom part. The bottom part is along of the oligos. So this mRNA will be captured by our oligos. Like you can see here, will be captured by these oligos. So once being captured, so let me see another. So once this let's take this photo as the 90-degree change, right? So falling down, this mRNA will be captured by these oligos. Then we will similar to the reaction in the surface of the gel beads. We will make the cDNA, and in the cDNA we will make the cDNA with the spatial barcode. That means this is the final sequence we need to sequence. The final fragment we need to sequence. Once we know the sequence, we know the spatial barcode. We can tell which location does this coming from.
Oh, I see. That means you have this interesting technology to have multiple labeling. One is spatial labeling, right?
Yes, spatial labeling.
In terms like IC design, we have address, multiplex.
Yes.
So that interesting. You have this kind of spatial barcode. That means it's address, right? That means spatially I have a coordinate system, and then at the same time you also have some information about data related to the sequence information. And this is a very, very interesting idea to put together. I see.
Yeah. So for the single cell service, the gel bead service, we call it the single cell barcode. But for the spatial method, we call it the spatial barcode. But they are very similar.
I see. Very interesting. This is I see. So of course, I understand. Another question. I know that there's in recent 1 or 2 years, there's a lot more people you look at your company product to look at those kind of tumor, right? Tumor has actually is very heterogeneous, so that in this case, if you spatially to do the encoding of those so many cells in your tumor, then so that I can see, so that you can really look at large number of cells from tumor to look at heterogeneous behavior, right? Oh, I see. So that's I think that's really the value of your company's product.
Yes, yes, you can see from this T-SNE plot, we can tell the different T or B cell just based on their marker, either the gene expression marker or the cell membrane marker or the attack method. So this is why we said the multi-omics way is very impressive because for some of the sample, we cannot separate them just based on their gene expression. But we can, once we integrate the protein expression, or we integrate the open region address, so we have more markers from different dimensions, we can better separate the cell type.
Okay. This is I think in terms of idea, it's quite very novel. I would say it's a really another important breakthrough from my point of view.
Yeah, and another information is I think you have heard about this digital droplet digital PCR.
Yeah.
When I was in Bio-Rad, you will tell that actually the microfluidic design, the microchip design is very similar to each other.
Yes, yeah.
Because the founder of 10X was the founder of that technology.
Oh, I see.
Yeah, so when I was in Bio-Rad, I see some photos because at that time we used the droplet to capture the DNA. One day I saw the photos from Bio-Rad R&D team. They said, okay, so I give you this very impressive photo. Can you see? It's the cell in the droplet.
I see.
So finally, 2 or 3 years later, we see this single cell product coming out.
I see.
Yeah. It's very interesting journey.
I see. So this microchip is made by what? Plastic or some glass or some plastic.
Yeah.
Okay. It probably is injection molding.
Yes.
To make it low cost. I see.
Yes.
But what's common? If you use glass or plastic, so of course plastic is much cheaper.
Yes.
I see.
And I also to reply that microwave question. Now we have there is one group in Zhejiang University, Professor Guo Guoji. They had 2 papers. They used PDMS to fabricate this microwave plate for the single cell analysis. They used the name called microwave-seq.
I see.
I heard they are also trying to commercialize that microwave-based technology.
Okay, I see. Okay, great. Okay, I don't know if any other question from their student or postdoctor, whatever. Lingzhen, do you have any question? Hello? Lingzhen?
Hi, Professor.
Yeah.
Hi, Dr. Lin. Thanks very much for your presentation. I just have a very simple question because I'm not majoring in bio, I'm majoring in electronics. I'm interested in the sequencing machine. You said you used the sequencing machine from Illumina. And my question is, what is the typical length of the sensing target? The sequencing of that sensing target.
Yeah, that's a very, very important question. When we talk about this sequencer, so Illumina now their maximum read length is 2 by 300. 2 by 300 base pair.
2 by 300.
Yes. So if we also count the adapter on the 2 terminals, they will be maximum, I think, 700 BP.
Okay.
BP, yes. But usually Illumina now, they majority of the instrument, they only support 2 by 150 maximum. So the fragment should be 350 to 400 BP maximum.
I see. Can you please go to the slide 20? Not this one, 21, please.
21, yes.
Yeah. So what I'm asking is the typical length of this sequence you want to sense a sequence here. What is the typical length?
Oh, you mean the length of this oligo, right?
Yes.
Yeah. The poly-DT 30 nucleotide, 30.
Okay.
And,
base pair.
Because it's single. Because it's a single.
Single. Okay.
Call it,
single.
Yeah,
okay.
NT, but it's the same idea actually.
Okay.
So 30 NT, and the UMI is 12. And the 10X barcode is 16. And the this part, I did not remember very clearly. I think between 12 to 20. Between 12 to 20. I don't know I don't remember the exact number.
Oh, I see.
Yeah.
So I think the length is not that long.
Yes.
Do you think if we have a portable like a sequencer like based on semiconductor technology, can we make this application even broader?
You mean make which one even broader?
I mean, can we make this? This application, if we can provide a portable device where we are like in any aspect, can we improve its its application, broaden its application range, or the other side? Yes,
yeah, it's a very good question. I think the what I say 10x our R&D or our founder is very smart because you can see firstly they just have one oligo on the membrane of the gel beads, so they can only do the gene expression. And then they have some good idea that we can make a very similar oligo, but just change the sequence here with some specifically designed sequence. So using this sequence, they can capture the guide RNA as in the CRISPR application, or they can capture the cell membrane antibody because the antibody label with that barcode. And also, I think T cell or B cell antigen special label. So I think, yeah, you just use the oligo thing, but change the frequency. Change the sequence. They can integrate multiple way into this. So as long as any feature can be captured by this oligo, that they can expand, I think.
I see.
Yeah, so that's why I say that they are very, very smart. They never change they don't need to change the physical size or the material of the gel beads. They just integrate different sequence for different use.
Okay. Thanks very much for your answer.
Yeah, hope I answered your question. Yeah.
No, I also want to ask the question about this thing. That means if somehow use this kind of gel bead, and then we have additional like engineering about this one, which maybe has some electrically, chemical, this kind of interface, maybe connect to, for example, like my postdoc Dr. Lin Zhen's this design, so that that may be generate another new branch of this technology.
Yes.
Electrochemically, seeing that for especially as I told you, right? So like in your new iPhone, like my iPhone 10, we have 7 billion of transistors, so that if somehow we can use this kind of your gel bead and then have some additional design, electrochemical probe or some or hybridize with our maybe 1 billion or 10 billion of this kind of sensor array, so that then to electrically I can really do another way for detection or something. I think, what do you think?
Yes. So,
but of course, that requires additional engineering on this kind of oligo, and then
so you can see that I think the gel bead size. I don't know the exact number, but I guess it should be 50 to 60.
I see.
Right. If this is a cell, this is
I see. Yeah. Okay. So means what do you think? Do you think so that we hope that we can develop something which compatible with 10x genomic, right? This is something then and then integrate microfluidic with our semiconductor, this kind of sensing platform, right? So that's something would be very interesting, right? Do you think? Means do you need anything, right? Do you think? So that the key thing, of course, maybe we should ask our NIGA to think about from chemical engineering point of view, how to have a new engineering about that oligo, right? So that additional electrochemical engineering and the pro, right? So to be compatible with this kind of semiconductor, this kind of sequencing platform or biosensing platform. Yeah. That would be very interesting. Yeah. Niga, do you have any question you want to ask?
No, before I also see on YouTube about this product. It's really interesting. And no, I don't have any question about
so how we try to do this kind of integration, right, between the this kind of 10x genomic, this kind of microfluidic with semiconductor, this kind of ISFET EGFT sensor done by Mingzhen and the other PhD student in my lab, right? Doing that. So I think that'll be very interesting way of thinking about this. Yeah.
Yes.
Because I do understand this kind of engineering is something I cannot do. Basically, I have very strong background in microfluidic mechanics, but that requires some special kind of engineering. This kind of the GOB.
Yeah.
Okay. Any other question from the other student? I see, huh? Probably we have a lot this kind of discussion, so that if not, probably we should thank Dr. Lin again. So that thank very much to spend time to give this presentation, and I think thank you very much. I think probably that's it for today's this kind of online seminar. Okay.
Yeah. Thank you for the invitation. And
Thank you,
Yeah, if you have further question, we can keep a discussion offline. Yeah, I'm very glad to give my help.
I see. Okay, so I think that's it. So okay, bye bye.
Bye bye.
Okay, thank you very much. Bye bye.